All right. Well, I'd like to welcome everyone to today's edition of the Polyploidy Webinar. This is the last Polyploidy Webinar of this academic year of 22-23. As we were just chatting, I think we'll probably keep doing this next year, but hopefully we can have a chat about that. And hopefully I'll see many of you at the Polyploidy conference happening in a month. So May 9th to 12th in Florida. This is the registration is open now. You can go to polyploidyconference.org or polyploidy23.org, I believe. No, it's polyploidyconference.org as I'm looking at it. Check that out. I'm going to be sending an email out actually to the mailing list later this afternoon with some details for registration. There's a wonderful deal. I think Doug and Pam Soltis did an amazing job getting not only the facilities for this, but also a wonderful deal on the room rate. So I encourage you to check that out. We'll all be there from my lab, and I know other folks that are heading there as well. And so the next, Polyploidy webinar, hypothetically, the next chat will be in 4 weeks at the conference there in Florida. So we'll be talking lots of polyploidy. Hopefully everybody can make it there. It looks like a great venue. And again, I'll post a link again, or I'll send an email out to everybody so that you can all have all those links and information and things about the early bird registration, the reduced registration for students and postdocs, which is very reasonable, and more to come. All right. And with that, I'd like to get started with our two talks today, which I'm looking forward to both of these. And up first is Dr. Sreetama Bhadra. She's a plant evolutionary biologist and cytogeneticist who's working as a postdoctoral researcher in the German Center for Integrative Biodiversity Research, or IDIV, at Hall Jena Leipzig, in Leipzig, Germany. And she's been there since 2021. And this follows a PhD at the University of Calcutta, India, as well as postdocs at the Central European Institute of Technology in the Czech Republic and the Chinese Academy of Sciences. As a scientist, her research focuses on understanding how polyploidy and genome size predisposes plants to trait variation that may impact their macroevolutionary trajectories and the macroecology of plants. And her work aims to assess the role of polyploidy, genome size, and traits in past adaptive and diversification of flowering plants, as well as identifying how those traits interact with their vulnerability to future climate change, and how that may contribute to more robust conservation of vulnerable species that may have less adaptive potential. She's authored multiple research articles in a variety of journals and presented her work in many different conferences, including here today at the Polyploidy webinar, where she's going to tell us about her work on unraveling the connection between genome size, trait flexibility, and the evolutionary radiation of angiosperms. With that, I'll let Dr. Sreetama take it away.
Thank you so much, Mike. So this project, the presentation for today, this is a part of my postdoc project here in IDIV. And so we have a huge collection of a huge amount of biodiversity right now on Earth, and it amounts to about 8.3 million species. But among these 8.3 million species, only a tiny fraction of them are plants, which is about 0.4 million species. But if we look closely, even within this 0.4 million species, a whooping 80% are angiosperms, and the rest 20% is occupied by all the other groups of plants. And it has always been a puzzle to understand why is this and how angiosperms became so dominant in the present day Earth. So this huge diversity of angiosperm has been attributed to evolutionary radiation, which is basically the simultaneous diversification of multiple lineages of angiosperms, and simultaneous diversification means speciation minus extinction. And this was perfectly exemplified in the study of 2019 by Soltis et al., where they showed that in this figure, where exactly these diversifications has taken place, or major radiations have taken place in these yellow stars. And one of the major aspects of evolutionary radiation is the evolution of appropriate traits. However, we should be cautious from the very beginning, because correlating evolution of appropriate trait only with evolutionary radiation is perhaps an oversimplification of a much more complex interaction of biological and environmental processes. However, even if we look at the traits only, it is mostly not one particular trait, but a trait syndrome or a group of traits that facilitate this type of radiation. Krebs and Nicholas in 2009 suggested that trait reinvention or repeated evolution of traits in angiosperms have actually helped angiosperms to occupy such a huge, to spread across the world. And along with this, Ornstein in 2019 suggested that along with this reinvention, the high evolvability or trait flexibility of angiosperm, that is the repeated and independent gain and loss or transition between traits and trait states and trait syndromes at the macroevolutionary scale, has resulted in this huge diversity. However, we are yet to actually see whether these hypotheses holds true, and we don't have much of a direct proof. And along with that, we also do not have a very clear idea about what is the genomic predisposition of these trait flexibility. And when we talk about genomic predisposition and traits, we tend to look at genes. However, single genes control specific pathways or mostly part of pathways, and expression of a trait does not really depend on an individual gene. So we have to have a more holistic view, and we have to zoom out, and maybe looking at the plant chromosome is a way out. And we already know that plant chromosome numbers have evolutionary significance, and this chromosome number varies mainly by in plants mainly by the whole genome duplication or polyploidization event that increases the chromosome number along with increasing the genome size. There is a total amount of DNA in a cell that increases the gene dosage, increase robustness of the phenotype, and create reproductive barrier and ecological isolation. In angiosperms specifically, along with this polyploidization, a beautiful event happens that is called post-polyploid diploidization over macroevolutionary time, that all these duplicated chromosomes undergo genome rearrangement that leads to diploidization or reverting them back towards a diploid-like state. This directly affects the genome size and reduces the genome size and decouples genome size from chromosomes, which has many examples. And this may also result in reduction in chromosome number, potential genetic novelty, and phenotypic changes. So chromosome numbers and ploidy level have variable phenotypic effect and may not be the right proxy to understand trait flexibility. However, when I was talking about chromosomes, one particular aspect or one particular property of DNA has, one genomic factor that came up constantly was the genome size. And genome size may and also have a very steady and stable effect on the phenotypic chain, on the phenotypic traits because of the physical mass, regardless of the information that is present in the genome. And this is called nucleotypic effect. And this actually regulates the minimum size a cell can obtain and also how much time a cell will take to complete a cell cycle. These together affect the anatomical traits, impacting the total number of cells and the distance between the cells, especially in case of stomatal size, vein density, which directly affects physiological characters like transpiration and photosynthesis. And also, it affects the minimum nutrient a plant requires because DNA is a very nitrogen and phosphorus demanding molecule, and a plant with large genome size will have a higher nutrient demand than a plant with a small genome size. And all these together affect the morphological characters. However, we really do not have a lot of study that have directly showed any correlation between genome size and morphological characters, except a few that has shown it on the relation between seed size and genome size. So genome size variation does not only occur by whole genome duplication and post-polyploid diploidization, it is also results from DNA repair mechanisms and repeat amplifications. And in angiosperms, there is a huge variation in genome size that has about 2,400-fold, which is a very large compared to their sister groups like gymnosperms with only 16-fold and pteridophyte with about 200-fold of variation. And so we would predict that genome size, so if there is in a lineage, there is a large genome size at the base of the lineage, there will be restricted trait flexibility or trait evolution because the plants will be pushed towards larger traits and there won't be as much of diversification. However, in case where the lineages, at the base of the lineage, if there is a small genome, it will have a higher trait flexibility, and these traits can change from small to large genome, which might result in a higher diversification. To test whether this is true or not, we took palms as a model system, and palms show very high trait variation, though Maybe when you think about palms, you mostly think about coconut or date palms, which has sort of a steady size. However, if you look closely on different palm species, it has, like, you can find you can find palms that are sessile, and also palms that has a stem length of about 60 meters. Also, palms have very different variation in leaf size and leaf types. They have pinnate leaf and palmate leaf, and also very large leaves of raffia, which reaches a height of 25 meters. Also, palms include very huge variation in seed size, where they have a very small gene of very small seeds, and also the largest seed that you can find in the world of the double coconut. And this high trait variation has resulted from trait flexibility. Palms also have quite a high range of genome size variation, which varies for about 60-fold. And this variation is not by an effect of any recent polyploidization. And this we can understand by the low chromosome number variation that we find in palms. And there are only 4 exceptions. There are 4 polyploids among 2,500 species. And palms are also have they are divided into very clear 5 subfamilies. So putting this together, taking the palms, we wanted to test 3 hypotheses. First, we hypothesized that the rates of diversification and rate of genome size and trait evolution have increased in the neogene that has helped in the establishment of the modern day vegetation. The second hypothesis that we have is the rate of diversification is dependent on genome size, with larger genomes constraining diversification rate, while similarly, the large genomes will also constrain the rate of genome size evolution and hence trait flexibility. Our third hypothesis is that high trait evolutionary rates or flexibility are associated with high diversification rate, because partitioning of ecological space due to high trait evolution rate may lead to speciation and hence diversification. To test these hypotheses, we collected data for diversification. We used Forby et al.'s data of 2016. For genome size evolution, we used this phylogenetic tree along with the genome size data that we got from our 2 collaborators. And for trait flexibility, we used this phylogenetic tree along with the trait data that we obtained from palm traits database. And we used the quantitative traits of fruit size, seed size, stem, and leaf size. And we used Bayesian statistics to model the phenotypic evolutionary rate and diversification rate across phylogenetic tree. And we used BAM for this study. And for BAM, we first have to analyze the data in C++ and then export this data into R and use BAM tools package in R to obtain something called a BAM data object, which is a complex data structure that has phylogenetic tree with map of all the macroevolutionary rate parameter. So here we extracted some specific things from this BAM data object. First, diversification rate of each species. Second, genome size evolution rate of each species. And third, the trait evolution rate of each species. So for all the audience, please bear with me, because when I will talk for the rest of my talk, if I say tip rate, if I say trait evolution, if I say trait flexibility, that would mean the same thing. And I have a tendency of mixing these terminology up. So first we visualized in a phylorate plot the diversification of palms. And here in this plot, the more red, the more red it shows the higher rate of diversification, and the more blue it shows a lower rate of diversification. And we actually found quite a lot of variation even within subfamily when it comes to the diversification rate. But overall, palms did show high diversification rate. Then we wanted to test our first hypothesis that how this palm's rate of evolution and diversification has changed over time. So we plotted this, plotted all the diversification rate and trait evolution rate and genome size evolution, and we did find that from around 20 million years ago, this diversification rate has increased, genome size rate evolution has increased, and there has been changes in the trait evolution rates too. So this actually proves our first hypothesis. And then we wanted to see whether the plants that has high diversification rate also has a high trait flexibility by some eye estimation for initial confirmation. So we plotted the genera that showed high diversification rate and checked what was the trait evolution rate in these genera, and many of them did show high trait flexibility individually. We also then wanted to check what are the the species with high trait like extremes of traits show in place of their own trait flexibility, but except Raffia, which did show high trait flexibility with high trait variation. Others did not really show high stem evolution rate or high seed evolution rate in case of Laurasia or Ceroxylon respectively. Then we wanted to do a multivariate model testing where we would put all the variables together and see what's going on among how they are affecting each other. But before that, we did a linear regression analysis. And for that, we needed to do this linear regression analysis because we wanted to avoid overestimation of correlation. For example, we find really strong correlation between fruit size and seed size. And if we had kept both these traits in our analysis, it could have underestimated relations, say, between fruits and stem. So this was a point when we actually eliminated our seed traits and kept our fruit traits in the further analysis. And also we wanted to understand the nature of this correlation, whether they will be positively correlated or negatively correlated. So then we did this multivariate model testing by structural equation modeling, or SEM, to test the influences among interacting variable, which combines the regression and confirmatory factor analysis. And we use the package Lavan. Briefly, the way this package works is we put all the variables, say here A, B, C, and D are 4 variables, and we first run and check if these relationships stand. And then we eliminate those variables which did not have a statistically significant value. And we rerun this model till we get a model that has all the relation with significant p-values. And we can have multiple models like this, and then we choose which is a better model by different fit indices. And this was the model that this was our final model that we got when we put in diversification rate, trait evolution rate, genome size evolution rate, and trait values together. And here the red arrows indicate the positive relationship, the blue values indicate the negative relationship. These are the fit indices that we got for this model, and the thickness of the arrows shows the estimate. So then we wanted to see what's going on with our hypothesis and what we got in our results. And for the first hypothesis, for this first hypothesis of hypothesis 2, there was like, we hypothesized that the rate of diversification will be dependent on genome size, with larger genome constraining diversification rate, but we did not really get that. We did not really find that larger genomes are constraining diversification rate in case of palms. Also, the second part said that larger genomes will constrain the rate of genome size evolution. However, we did get this one, that larger genomes of palms did constrain the rate of evolution of genome size evolution of palms. Coming to our third hypothesis, where we predicted that the high trait evolution rates or trait flexibility will be associated with high diversification rate, we did find that trait flexibility affected diversification rate. So summing these together, these are our conclusions from this part of the study, is that genome size evolution and trait evolution or trait flexibility did aid in the diversification and distribution of palms, leading to the establishment of tropical rainforest in present times, because palms are a major keystone species in these rainforests. Diversification of palms did increase around 20 million years ago, which corresponded with the increased rate of genome size, fruit size, and seed size evolution, while the rate of stem size evolution slowed around after this time. And genome size evolution affected diversification, but not directly. Rather, genome size affect trait flexibility, which in turn affect diversification rate. But even when we are looking at how genome size evolution is affecting trait flexibility, genome size do not affect all the traits, all the evolution of all the traits. Like, we did not see any relation between genome size evolution and leaf evolution. And among the traits that genome size affected, only stem evolution affected diversification rate in case of palms. So it is not always necessary that all trait flexibility will lead to changes in diversification rate. And large genomes did show constraint for genome size evolution, but not the diversification rate of palms. And the fact, which was a bit surprising for us, that we did not find any effect of leaf in this whole study, in this whole model. It might also be because palm leaves are affected by abiotic factors like temperature, which we have not yet considered in our study. And this might be a reason why leaf traits got eliminated from the whole study. And beyond this, now I'm also trying to understand how these trait flexibility and diversification rate and genome size evolution rate vary across different biogeographic regions. And I will continue the study to include the rosids to evaluate our results more generally. And with that, I would like to thank all my collaborators and co-PIs, especially Renske and Ilya, who are the co-PIs of the project, and our amazing SDiff team and from IDIF, and William Becker and Sidoni for being the collaborator of this project. Thank you.
Well, thank you so much, Sreetama. That was a wonderful talk. I have a couple of questions, but I'll go to the chat first and see if anybody wants to put their answer in the chat or raise their hand and ask on video. Usually takes everybody a couple of seconds. I'll go. I see. I'm going to go ahead and jump in here real quick and ask a question. So when you showed the plot, it looked like that there was a positive correlation between genome size, the rate of genome size evolution, and stem height. Was that true? I was trying to parse that out. Okay. Yeah, that big blue. There it is.
Yeah.
Interesting. That's cool. So is that? I'm just thinking about the water relations here. So I was trying to think about, like, so does this suggest that there's some, I think, with the genome size and the cell size and just with how you get a tall, a tall plant, is this because that you see like really flexible genomes, like a lot of variation then in genome size evolutionary rate, or is this that I'm trying to understand is that you see that the rate of genome size increase, is it a unidirectional sort of value there is correlated with stem height? And when stem heights are you talking about I don't know, is this some kind of interaction then, I guess, between changing genome sizes to match cell sizes to kind of adjust plant heights, or yeah, maybe can you expand on that a little bit?
Yeah, I mean, yes, this is one
as
thing
much as you can.
yeah, sure. Because this is one thing that I also feel that we need to include other factors to clearly understand what exactly is going on. But yeah, I mean, when I'm saying that genome size evolution is like, affecting genome size evolution positively, negatively, it is not growing this size change from small to big.
Right. Yeah. It's just
the variation
in that trait itself. Yeah.
Yes. Yeah. It's a variation. It's not like unidirectional variation. It can be on both sides.
Yeah.
And definitely, I think, yes, I mean, climate definitely plays a role. And also there are palms which are growing on the extremes of climate. So yeah, I think we have to do a bit more of research, literature search, and analysis to understand what are the other factors that might be involved.
Yeah. It just seems like a really interesting relationship because the just the physiology, effects of changing the size of the xylem and phloem cells, right? And how that influences conductance and height. And there's this, a lot of that's beautifully constrained in a lot of trees, but palms come in all sorts of shapes and sizes.
Yes.
And so
also, one unique thing about palms. Yeah, one unique thing about palm is they are monocots. They are not dicots.
Right.
So even like we did not put in a direct relation between stem diameter and stem height, because they don't have really a proper secondary.
Exactly. Yeah.
So this sort of things get complicated when it comes to palms.
But it's an interesting relationship that anyway, I love that. Thank you for that. And it seems like there's a lot of really cool follow-up work to do there. So.
Mm-hmm.
All right. There's a question in the chat from Rob Laporte. Rob, if you want to hop on video, and feel free to ask.
Yeah. Thanks. It's coming up, I guess, maybe. Thanks, Sreetama. That was great. I think you kind of addressed this a little bit, or I guess Mike was sort of dancing around this too, but with these climate changes and the effects here, I'm wondering to what degree some of this mid-Miocene climatic changes might have influenced this. It doesn't seem like you have that in your model, and I don't expect you to at all, but like, at that time, we see the expansion of all these grasslands. It's cooling and drying out generally around the world. So I'm curious about how this diversification might correlate with some of that, and whether you can sort of extricate that from these other changes that are happening globally. I mean, it just seems like there's all these changes that kind of are just contributing to this rapid phenotypic evolution, but it's wrapped up with all these other sort of like biome and ecological changes that are occurring at the same time.
Yes, sure. Yeah, I think for definitely to some extent, this climatic changes affected for sure. But another interesting thing about palms is they are tropical. They cannot grow in the temperate region. So it's really a bit tough to see how, like, if the cooling happens like really fast, how that would ultimately affect, because it will be a problem for their survival. So yeah, but definitely, I mean, the time after KPG and this in around the Miocene, I mean, this is how they like stabilized, right, around this time. So yeah, thank you.
Yeah, thanks. I mean, it's just an interesting group to think about anyway, but correlated with all these other forest changes and grasslands expanding into even the sort of lower mid latitudes. I think it's just a fascinating system. So cool. Thanks.
All right. Well, if there are no other questions for Sreetama, we'll get on to our next talk. And if anybody has any questions, feel free to save those until the end. We'll have a chance to ask them then. All right. And we'll let Nélida load up her slides next. Right. Hopefully you can share. All right. With that, looks good. And we'll get going here. Their second talk of the day from Nélida Padilla-Garcia. Nélida is originally from the south of Spain, where she studied and earned her degree in a master's in biology, where she became fascinated about plant evolution, and subsequently went to pursue a double PhD at the University of Salamanca in collaboration with the Museum National d'Histoire Naturelle in Paris. And I know I'm butchering all of these names. And then after finishing her PhD, she moved to the Czech Republic in 2020, where she is a postdoctoral researcher working at Charles University in Prague. And she has recently found out she'll be received a position where she'll be starting a tenure track position at the University of Madrid next month. So congratulations to Nélida on that new position. She is broadly interested in plant evolution and the adaptive value of evolutionary processes in natural populations. In particular, she's interested in the role of polyploidy and hybridization and how it contributes to ecological adaptation and speciation. Her work combines extensive field sampling with flow cytometry, ecological, and next-gen sequencing data to study ecological adaptation and plant speciation. And her current research focuses on population genomic studies in Arabidopsis aeruginosa and Lyrata. And the work she'll present today is some of her postdoctoral research from her time in the Department of Botany at Charles University with Philip Kolar and his team. And she'll be telling us about niche differentiation following whole genome duplication and interploidy introgression. With that, I'll let Nélida take it away.
Thank you, Mike, for the introduction and also for organizing this series of webinars. I'm very glad to participate on that. So today, the title of my talk is a question, indeed. And I would like to tell you about the work that I have been doing, trying to address these questions. So if it's niche differentiation after whole genome duplication and interploidy introgression. And as Mike said, I performed this work at Charles University in Prague, where I have been working in Philip Kolar's lab, but also in collaboration with Karol Marchol and his team. So I will start with a very, very short introduction because, as you all know, polyploidy or whole genome duplication is a leading evolutionary force for plant speciation and diversification. It is a widespread phenomenon, and it has been estimated that almost 15% of speciation events in angiosperm are associated with some polyploidy increase. Polyploidy has been also considered a mechanism of sympatric speciation. And this idea has been mainly reinforced by the post-zygotic reproductive isolation that diploids and polyploids potentially experience immediately after the formation of polyploids, due, for example, to the difference in number of sets of chromosomes. However, we also know that these post-zygotic barriers, as the triploid block or the reduction of thickness of triploids, could be incomplete. So we all agree that polyploid speciation is not an instantaneous process, and that other prezygotic reproductive barriers can also play a role in assortative matting between diploids and polyploids, and consequently in polyploid speciation. So one of many of these prezygotic barriers which can be important for the establishment of neopolyploids is habitat differentiation. Niche differentiation is generally associated with polyploidy due to the novel phenotypic, physiological, and genetic combinations that can arise after whole genome duplication. And this novelty could potentially lead to polyploids to expand to new ecological niches that remain maybe unexplored for diploids. It's true also that this pattern might be biased towards allopolyploids that arise from two different genomes, and thus they show higher heterozygosity than autopolyploids. However, the existence of niche differentiation between diploids and autotetraploids really differs between species. So if there is something that we have learned after many studies on this field, is that there is no universal pattern about the role of niche differentiation in polyploid establishment and evolution. And one of the explanations to this inconsistency is that niche evolution might be detectable only when polyploid lineages are compared with their closest diploid ancestor, something that is not always done. Moreover, recent studies has also shown that polyploid barrier is permeable. So hybridization and introgression can occur between diploids and polyploids more often than expected. And this introgression might be also adaptive. For example, if this introgression or genetic exchange between diploids and polyploids provide some beneficial variants to polyploids that can allow them to colonize new ecological niches. So in this project, the main general questions I am interested in are, first, which is the impact of the evolutionary history of the species when we are testing for autopolyploid niche evolution. Second, how strong is interploidy introgression in natural populations of autopolyploids? And also, in third, which are the consequences for adaptation of polyploids to new habitats, the consequences of this interploidy introgression. To answer these questions, we use Arabidopsis arenosa as a model for our study. And Arabidopsis arenosa has been a model system for already a large number of genomic studies on adaptation. And I think it's very important to contextualize first the study understanding the biographic history of the species. So Arabidopsis arenosa is a diploid autotetraploid species. Plants are perennial and obligate outcrossers. In this map, you can see the distribution of diploids represented in red and tetraploid populations represented in blue for the whole geographical range of the species, which is mainly Europe. And the center of diversification is the Western Carpathians in the Central Europe. In a previous study, Philip and collaborators identified a total of 5 diploid lineages within the species, distributed mainly in Central Europe, but also you can see with some diploid populations located in the Baltic Sea coast. Autotetraploids of Arabidopsis arenosa originated only once. So in the Western Carpathians, around 20 or 30,000 generations ago. And from there, tetraploids subsequently spread through Europe. And one of my first objectives when starting this project was to investigate which was the genetic structure within these tetraploid populations. And we identified 5 additional tetraploid lineage that were geographically separated in different areas of Central and Northern Europe. Previous study also have shown some evidence for interploidy introgression in secondary contact zones, mostly in the direction from diploids into tetraploids. But how strong this interploidy gene flow is and if it's related or not to niche divergence, it's remained unknown. So in order to address the first question, I tested the following hypothesis, whether the niche shift of polyploids was detectable only when polyploids were compared with their corresponding ancestor and not globally. For this niche comparison analysis, I use 352 localities, including both diploid and tetraploid cytotypes that we were able to assign to the intraspecific genetic lineage identified in Arabidopsis arenosa. The environmental data that we used was extracted from WorldClim, and after evaluating the correlations of all the variables, we use 9 not highly correlated for the analysis. These variables were related to temperature and to precipitation. We quantified and compared climatic niches using an environmental PCA and running different tests for niche overlap, equivalence, and similarity. We also quantified niche dynamics using different indices, and we also calculated the niche breadth for each of the lineage. The variation that was explained by the first 2 axes of the PCA was 39 and 29.5% respectively, which is quite high. And for all the graphs I am going to show you today, the niche of diploids will be always represented in pink color. The niche of tetraploids will be represented in blue. And when there is a niche overlap between both, will be shown in purple. So in this case, what it is represented is the comparison of the niche of all diploids and tetraploids when they were compared globally. That means without considering the assignment to the intraspecific genetic lineages. And we did not observe niche differentiation between diploids and tetraploids when doing in that way globally. However, we found very different patterns of climatic niche evolution when we compare each tetraploid lineage of Arabidopsis arenosa independently to the niche of the ancestral diploid lineage from the Western Carpathians, where polyploids originated. Specifically, we found niche conservatism when comparing diploid and tetraploids from the area where tetraploids originated in the Western Carpathian. So this result confirms that autopolyploids and diploids progenitors can coexist in the same niche. But in contrast to that, when tetraploids from Alpine, rural, and Central European lineages were compared to the ancestral diploid progenitor, we observed that they have experienced niche expansion compared to the diploid ancestor. And last, we also observe niche contractions of tetraploids when tetraploids from Southern Carpathian were compared to the diploid ancestor. So overall, this study confirms that niche evolution of polyploids in Arenosa was only detected when we assigned the populations to intraspecific genetic lineages, not when we were comparing the niche of diploids and tetraploids globally. And these results also demonstrate that the niche shifts of tetraploids in Arabidopsis arenosa is not only driven by whole genome duplication, but probably reflects other evolutionary processes that occur after the whole genome duplication event in the Western Carpathian. So to be able to address the second main question, so how strong is interploidy introgression in natural populations of Arabidopsis arenosa, we set an experimental design consisting on 4 contact zones where diploid and tetraploids coexist. One is the primary contact zones where tetraploids originated, and 3 additional secondary contact zones found in the Dinaric Alps, the Southern Carpathian, and the Baltic Sea coast. I just wanted to mention that individuals of intermediate ploidy, that means triploids, are very rare in nature, so we did not include them in our analysis. Here, I have represented the populations that we selected for the study. So we sequenced 8 individuals per population. And in each contact zones, we have 2 types of populations, what we call single ploidy populations. These ones that are comprising only one cytotype, that means diploid or tetraploids, and also mixed ploidy populations, where diploids and tetraploids are coexisting next to each other. For example, this is a picture of a mixed ploidy population in which you can observe that diploids and tetraploids individuals are intermixed, so very, very next to each other, while in single ploidy populations, diploid and tetraploid could be apart from each other about 20 or 30 kilometers. So using different combinations of these populations, we calculated whole genome D statistic to test for introgression between diploids and tetraploid individuals. And we found significant evidence for this introgression in all contact zones with a higher admixture proportions when we compare diploids and tetraploids from mixed ploidy population than with a single than when we compare it from single ploidy population, what it was our expectation. So having this strong, or observing this strong interploidy introgression in Arabidopsis arenosa, we move to our last question. So which are the consequences of this introgression when testing climatic niche evolution? The expectation for such interploidy gene flow are similar, could be similar to the hybrid origin of allopolyploids, because for tetraploids, introgression in secondary contact zones with other diploid lineage that are locally adapted can increase the phenotypic and genetic variation and also the chances for them to adapt to the new ecological conditions. So to test for this effect, we use an ecological niche comparison approach by adding the niche of the diploid sympatric donor to the analysis and testing if it helps or not to explain the niche of the tetraploid lineage. We tested this hypothesis for now in 2 contact zones where the tetraploid lineage is introgressed by diploids. So it's in the Southern Carpathian and in the Baltic coast. And the results, once again, were not the same for each contact zone. So first, in the Southern Carpathian, when we added the niche, you can see in this square in red. So when we add the niche requirements of the sympatric diploid lineage from the Southern Carpathian, we did not observe any substantial changes in terms of niche expansion, stability, and unfilling indices with respect to the previous comparison here in gray, when we include only the ancestral diploid progenitor, that is the Western Carpathian diploid lineage. And as in a similar way, in this new analysis, a considerable proportion of the niche of the diploid lineage has not been filled by the tetraploids, indicating that probably this introgression did not lead to a broader niche of the tetraploid lineage in the Southern Carpathian. But on the other hand, we observe here also signal in red, we observe a lower value of the niche expansion index and also a lower value of the unfilling index when adding the niche of the diploid Baltic lineage, the one that was locally adaptive, to the analysis with respect to the previous comparison when only the ancestral diploid was included. So we can interpret that introgression of the Baltic diploid lineage into the tetraploid lineage has contributed to the ecological niche of the tetraploid. So to conclude, I just would like to highlight 2 messages from the talk. So the first one is that we need to take into account the evolutionary history of polyploids when testing for niche differentiation. And the second is that interploidy introgression is probably a very important process for ecological adaptation of plants to new environments. And I have some work also in progress to investigate more deeply about these effects of interploidy introgression in adaptation of tetraploids of Arabidopsis arenosa. So stay connected. I hope some new results will come soon. So with this, I just want to thank all the collaborators on the project, and also all people from the team helping with the collection of samples and also with bioinformatic analysis, and you for being here today. And I'm very happy to answer your questions. Thank you.
All right. Well, thank you, Nélida, for a wonderful talk. And this is such a great paper, too, if people haven't read it yet. So are there any questions? If anybody has any questions for Nélida, feel free to pop them in the chat or raise your hand and pop your video on. Yes. Yep. Really, really, really cool work. So, Rob, do you have a question? Sorry, I was going to ask something myself, but go ahead.
I don't specifically, and I have to run off to class right now. But yeah, thank you, Nélida. That was really cool. I mean, I think it's really interesting to see some work like that showing some of the evolutionary history and the dependency of the formation of some of those polyploids on the traits that they bring along, or at least some of the genetic background that predisposes them to some of the traits that are expressed in those environments. And it's nice to see that there is some of that dependence. I mean, I think we've speculated on it for a long time and had some hints of it, but it's really nice to see that paper. So thanks. I have to run, but thank you.
Thank you. Thank you very much, Robert.
Thank you. Yeah, I had a real quick question too, just thinking about this. Okay, Justin Conover had a question here. I'm gonna go ahead and ask mine real quick, and then we'll let Justin ask his. So do you think that if you were to replicate this in another sort of species complex, one of the things that struck me was thinking about how the starting variation of those diploid populations themselves may be important. So if you had, for the success of the polyploid species, so do you think that more diverse diploid starting populations might be an important criterion then for, if you think about those, essentially, are you inheriting the trajectory? Your study suggests you're inheriting to some extent the trajectory of your founders. So if the founders are on a good trajectory, are you on a better trajectory as a polyploid? Is that something we can take away from this?
Yeah, it's a great question. Interesting question, because it's true that theoretically, the expectation would be more diverse, more successful, right. If there is some introgression. But we are looking on that more on the introgression regions, more in deeply on the genome. And it's true that we cannot use for this kind of analysis if the population has not a good diversity, or
it's
if
not
some
differentiated already.
Effects has been. No, we are not exploring this, but it's a good question. Probably what we are expecting also, if it's when the diploid is better adapted, it can bring some.
It can bring those adaptive alleles,
potential to the tetraploids. But if this is correlated to diversity or not, that's really.
It seems like you've got such a great set of questions here and system to study this, and then there's so many other Arabidopsis and other brassicas or other things of the Brassicaceae as well.
Probably it's something to explore in the future.
It's wonderful. It's really fascinating. I'm gonna let the Justins go next. So Justin Conover was first, and Justin's got no camera. But Justin, do you want to pop on the video or the audio, excuse me, or do you want me to go ahead and ask that, Justin Conover? I can ask it, I guess. No mic either. All right. So Justin Conover asks, how do you differentiate interploidy introgression from isolation by distance in the mixed ploidy populations?
Yeah, that's also a good question. But in mixed ploidy population, I'm not sure if there should be this isolation by distance affecting, because it's really like meters of distance. So it will be probably something affecting when comparing single ploidy population, or I can see an effect more on that. So what we try to follow, it's a design in which we have replicates. So we are not testing only with one or not two population combinations, but we have several ones and in different contact zones. So I cannot see that some specific effect is driven by isolation by distance, for example, in one population, but not in all. No, I think the design could be good for this, but we have not anything very, very specific to differentiate from for now.
All right, I think that answered Justin's question. So Justin Ramsey up next. Justin, do you want to pop on your audio and video, or you need to read it off? There you go.
Sure.
Always good to see you. So.
Yes, thanks. Great talk. I just was curious if what's known about sort of the mechanism of introgression and if it goes both ways, or if that's known yet.
Yeah, that's a very good question. So we know experimentally that no diploids, so there is no triploids, no? As I mentioned, there is no triploids in the field, no? In nature is super difficult. So without triploids, we don't see how tetraploids can introgress into diploids. It should be a triploid. So from what, from what we know from the species, probably unreduced gamete formation, it's the more probable mechanism for introgression. So that is from diploids to tetraploids. But I am exploring some analysis in order to try to test this direction of introgression. So, but difficult to address, I have to say. But still trying to run some specific tests on that.
No, that's really interesting. It's also, I mean, the fact that if there are no triploids being formed, the dynamics will be one way, whereas if the mechanism of introgression was different, it could be both ways. So the biology of the system could really affect what happens in terms of dynamics.
And the fact is that triplets can be formed, at least experimentally, in the greenhouse, and also so that we didn't found it in the field. It doesn't completely discard that triplets are doing, are having some role. So, and when there are triploids, they are not sterile. They are able to reproduce, at least not experimentally. So this is why we are not still wanted to explore a little bit more about that mechanism.
Yeah. Well, great system. Thank you.
Thanks.
Yeah, thank you. That's really interesting to think about. And how that flows, and on that same topic, Allison Scott here has another question in the chat about hybrids here, and I'll let Allison pop on and ask.
Thanks. Great talk, Nell. And it's really cool to see these results and everything. I'm wondering about the role of interspecies introgression. My European Lyrata and Arenosa knowledge is not that broad, except that I do know that the tetraploids can cross here. And so I wonder if, because there's the one origin in Arenosa, and then there's multiple origins putatively in Lyrata. And so I just wonder if there are, of these like diverse now tetraploid Arenosa lineages or populations, do you see increased introgression in any of them with Lyrata, or can you see? I don't know.
Yeah, very good question. You are right, and you are familiar with the system. So yeah, there are introgression occurring between diploids and tetraploids, but what we did in our design was to avoid these areas, because it's very, very specific areas where Arenosa and Lyrata introgress, and we avoid this, trying to see which is the interploidy introgression within Arenosa.
Separate from this interspecies. Okay,
but
very
obviously, it is a very interesting question later, if how it can affect the introgression from Lyrata. But this is a different question. Yeah.
Okay, cool. Thanks.
Thanks.
All right. Well, are there any other questions for either Nélida or Sreetama today? I don't think so. All right. Well, I'd like to thank everyone for joining us, and this is the last webinar of the semester, and thank you to everyone else as well who spoke over this year. I think we'll probably do this again next year, and send out some invites, get that organized over the summer, and probably stick with this format of once a month with a couple of talks in this sort of quasi post-pandemic world that we're in now. But I love these, and we didn't get it organized in the fall, and there was a lot of people emailing me, so it seems like there's interest in keeping this going. So we'll post the open submission or the open sign up sheet here soon. If anybody is interested in participating, feel free to shoot me an email. We just try to make sure that we cover the diversity of research and places and peoples and everything that to sort of get a great perspective on everything that's happening in our community, and bring us all together. So and so happy to have anybody else volunteer. And if you've already given one, we've been doing this for a few years now. We've had, I think, 70 or 75 talks, so we can cycle back around as well at this point. But again, I'd like to also just highlight too, there's the big Polyploid conference here, May 9th to the 12th in Florida. At polyploidconference.org is the website to check out. Registration is pretty cheap for everybody, especially for postdocs and grad students. I think it's $50, and the room rate is also fantastic, cheaper than Airbnb when I was looking at it. So, and that's hard to beat. So, it'd be great to see folks there, and hopefully, yeah, you can keep joining us next year. And again, congratulations to Nélida for her new position starting soon. Interested in this work, I'm sure she'll be looking for grad students and postdocs in the next year. And thank you to both Nélida and Sreetama for wonderful talks today.
Thank you so much for organizing.
Yes.
Thank you, everybody. Have a great afternoon or a great evening, depending where you are.
Thank you. Bye-bye.
Bye.
Bye.